Okay, hello to all of you. So let's discuss chapter number 3. Let me this mode. So basically, in chapter number 3, we are going to talk about what amino acids are, what is the structure, and we are going to talk about how amino acids they link with each other, and how do they form polypeptides, and basically how the polypeptides um, they basically how they fold and how they attain secondary structures, how do they attain quaternary structure, tertiary structure, and then the quaternary structure. So this is all chapter number 3 and 4. So in chapter number 3, we are going to basically focus on the amino acids. So first of all, what is the amino acid? So proteins, they are basically macromolecules. So there are four macromolecules which make the cells. Or we can also call them biomolecules, or we also call them molecules of the life, like lipids, carbohydrates, proteins, and nucleic acids. So we will be talking about all these biomolecules in this course. So the first biomolecule we have started with is the proteins. So they are macromolecules, they're big molecules. They can also be called biomolecules because they're molecules of life. Why they're called macromolecules? Because they're big molecules, they're polymers. Now, what is a polymer? Polymer is something which is made of repetitive subunits called monomers, and in different biomolecules, there are different monomers. Like proteins, their monomer is amino acids. Carbohydrates, their monomer is simple sugars, and nucleic acids, their monomer is nucleotides. And lipids, they are macromolecules, the big molecules, the biomolecules, but they are not polymers. They're not polymers. They're not made of repetitive subunits. They are made of few things. We will discuss when we will be reaching to those chapters. So the very first macromolecule I'm starting is the proteins, and the monomer of a protein is amino acid. So first of all, we need to know what is the basic structure of an amino acid, and there are 20 amino acids. We will be talking about those amino acids, and every amino acid has a basic structure. So if we look here, you can see here, this is the basic structure of an amino acid. There is a carbon, and you must be knowing the valency or the bonding capacity of carbon is 4. So carbon can be linked to 4 different atoms or 4 different functional groups. So basically, carbon is in the amino acid is linked to one group called amino group and a carboxyl group, and it is also linked to hydrogen. And then there is a R side chain, or there's a side group which we can see. So every amino acid has the same basic structure. Every amino acid has an amino group, every amino acid has a carboxyl group and has a hydrogen. So what makes all these 20 amino acids different from each other? It is that R group which makes them different from each other. In all the 20 different amino acids, this R group is different. So amino acid is called, why it's called amino acid? It is called amino acid because of the presence of this amino group as well as the presence of the carboxyl group. Now carboxyl group is acidic in nature, which means this group is actually COOH. COOH. Right now it is in the ionized form in proton. That's why it's written as COOH group. So it is, COOH group is a carboxyl group, and it is a donor of protons. It's a donor of hydrogen ions. That is why it is acidic in nature. So this group is acidic in nature. That is why this amino acid, which is a subunit of a protein, has been, okay, sorry, so this is what is I just explained. Okay, so for every amino acid, I think I didn't tell this point, that every amino acid can exist in the form of two stereoisomers. One is the L form and the second is the D form. It's all on the basis of their configuration in the space, and the molecules which differ from each other in the configuration in space, they're called stereoisomers. So the amino acids, they can exist in L and D forms. So I think I have explained this basically. You can go over this. And the carbon, because in the amino acids, the carbon which is in the center, to which amino group and carboxyl group and the R side chain is attached, and the fourth atom is the hydrogen. um, so this carbon is called chiral carbon, or it is called asymmetric carbon. Asymmetric carbon or the chiral carbon is that carbon in which the four groups or the four atoms which are attached to carbon, they're all different. Then that carbon is called chiral carbon. So the in amino acids, this carbon is called chiral carbon, or it's called asymmetric carbon, because these groups which are attached to this carbon are different, except only one amino acid which is called glycine, because in glycine the R group is itself H, it's itself hydrogen. So because there's a hydrogen here also, there's a hydrogen here also, this carbon cannot be called chiral carbon in the amino acid glycine. So that is the only exception. Other than that, all the 19 amino acids, they have the chiral carbon in them. So these are the 20 different amino acids: alanine, arginine, asparagine, aspartic acid, cysteine. This is the three-letter abbreviation, and basically this is the one-letter abbreviation for the amino acids. So what you need to know about these amino acids is we can classify these amino acids into four different categories. So there are amino acids which are non-polar amino acids, and then we have a second category of polar amino acids. Then we have a category of negatively charged amino acids. Then we have a category of positively charged amino acids. So basically, you need to know which amino acids fall in which category and why. What kind of, so as I said, all amino acids, they have the same basic structure. What is different is the R group. So R group is different in all amino acids. So basically, you need to know which R group is there in each amino acid. And because of the presence of the specific R group, because of the presence of the specific R group, the amino acids fall in different categories. So when the amino acids have positively charged R groups, they are positive amino acids. When they have negatively charged amino R groups, they fall in the category of negatively charged amino acids. So it all depends on the kind of R group they have. On the basis of that, they fall into different categories. Like for example, the first category is non-polar amino acids. So in this category are the amino acids that have non-polar side chains: glycine, alanine, valine, leucine, isoleucine, proline, phenylalanine, tryptophan, methionine. They all have non-polar R groups. So that is why, and here is the information given which R group specifically they have. And this is again the information about these amino acids which are considered non-polar on the basis of the R groups. So all these highlighted portions are the R groups. So it's the information is given here. You can go over it and you need to. Okay, so here it is shown that glycine, which is having the R group as H. So this carbon is not a chiral carbon. So this is the amino acid valine, and this is the R group which is present in the valine. So you can see this is all non-polar. That is why this amino acid is falling in the category of non-polar amino acids. This is alanine. Its R group is CH3, which is a methyl group. Methyl group is all non-polar. I hope you understand what is polar and non-polar. So polar groups are those groups in which, like, there is a unequal distribution of the electrons. So usually the groups which have oxygen in them or nitrogen in them, those groups are usually polar in nature, because oxygen and nitrogen, these are electronegative atoms, and they have a tendency to pull the electrons towards themselves. And as a result of that, there is an unequal distribution of the electrons, because when oxygen is forming a bond with some other atom, or nitrogen is forming a bond with some other atom, there is a tendency of these electronegative atoms to pull the electrons towards itself. But if the group, like this is CH3 group, you can see it's carbon and hydrogen, there is no electronegative atom here. So this, because this group does not have electronegative atoms in them, as a result of that, there is an equal distribution of electrons between these atoms because these atoms are forming bonds with each other. So that is why these groups are non-polar in nature. These groups are non-polar in nature. So that is why these amino acids are non-polar. So the second category is polar neutral amino acids. So this is the category in which includes your serine, threonine, tyrosine, cysteine, glutamine, and asparagine. So they, like for example, serine and threonine, they have a polar group like OH group. Now it has oxygen in it. So because of the presence of oxygen, which is an electronegative atom, this group is polar in nature, and this group is present in these amino acids. This group is present in these amino acids, and because of the presence of this group in these amino acids are polar. They are neutral, which means they have an unequal distribution of charges, but they do not have some overall charge on them. So you should know these amino acids also which fall in this category. This is again showing you all the polar uncharged amino acids, which include your threonine, serine, asparagine, cysteine, tyrosine, and glutamine. So you need to know all these things. And then the next category is basically negatively charged amino acids. In this, it's easy to remember. There are two amino acids, glutamic acid and aspartic acid. Their R group has a group called COOH group, which is a carboxyl group, which is I just discussed, which is also a part of every amino acid. Every amino acid has an amino group as well as carboxyl group. But these two amino acids, they have this carboxyl group in their R group also. So that is why these amino acids, they are negatively charged, because carboxyl group can lose is a donor of protons, and as a result of that, carboxyl group gets the negative charge on itself. So that is why these amino acids belong to this category. So this is your aspartic acid. You can see here, this is the R group, there is a COH group, this is glutamic acid, this is the R group, and there is a carboxyl group here. Then the next are the basic amino acids. These are basically the amino acids which have amino acids. This category includes those amino acids which have a positive charge on them. It includes histidine, lysine, and arginine. There are three amino acids which have positive charge on them. And why do they have positive charge on them? Because their R groups has some functional groups which give them the positive charge. So lysine, the side chain amino group, is attached to an aliphatic hydrocarbon chain. So in the R group, in the lysine, they have a amino group. So you can go over these. So this is basically showing you, this is your histidine, this is the R group, and this has because of this R group, it has a positive charge on it. This is your lysine. You can see this is an amino group, and because of it has a positive charge. This is arginine. This is a amino group which has a positive charge on it. So here positive charge, positive charge, positive charge. So they have some functional groups in the R groups because of which they have the positive charge. And next thing is, as I said, the amino acids are the monomers of proteins or the peptides. Now, a very, very important character of these macromolecules is, as I said, they are polymers and they are made of monomers. And these monomers, they are linked to each other. So how they are linked to each other? These monomers, they are linked to each other by bonds called covalent bonds. So covalent bonds, you must be knowing, they are very, very strong bonds. So these monomers, they are linked to each other by covalent bonds. And how the covalent bonds are formed? Covalent bonds are formed by the reactions called dehydration reactions. Okay, so basically this is one amino acid, this is second amino acid, so two amino acids. So together they lose a molecule of water. Okay, so basically COOH group is going to lose OH, and the NH, the amino group, is going to lose H, and together they are going to lose molecule of water, and that is going to link these two amino acids with each other by the formation of the peptide bond. So the peptide bond is actually formed between the carbon of the carboxyl group and the nitrogen of the amino group of the second amino acid, and that results in the formation of the peptide group. And as a result of that, these amino acids, they are linked to each other. So here it's shown here. So this is the peptide group. Okay, so this is one amino acid, So you can see this is the carbon, this is the hydrogen, this is the amino group. So this is the carbon, this is the hydrogen, this is the amino group, this is the carboxyl group, and this is the R group. This is the second amino acid. This is second amino acid. This is the amino group. This is the carboxyl group. This is the R group. This is the hydrogen group. So now this is one amino acid. This is second amino acid. Okay, this is one amino acid. This is second amino acid, and these two amino acids, they are linked to each other by peptide bond, and the peptide bond is formed between carbon of one amino acid and nitrogen of the second amino acid. So this is a peptide bond. So between all these amino acids, there are peptide bonds, and these amino acids, they are linked. These amino acids, they are linked to each other by the. These amino acids are linked to each other by the peptide bonds, and that's the way amino acids are linked to each other, and they form a polypeptide chain. Okay, so that's the way these amino acids, they are linked to each other. So that's all in this chapter, and in the next chapter we are going to talk about how these chain folds and acquires a secondary structure, and then the further chain folds more and form tertiary structure. So all about, so this is basically what we have just discussed is the primary structure of the protein. So the structure of the protein is at 4 different levels. That is what I'm going to discuss with you guys in the next chapter number 4.